Now, you have got a new drug that's an anti-estrogen drug. Let me ask you, why did we need an anti-estrogen drug, and what is the reason for the study that you're talking about here at San Antonio?
Well, it's not a classic anti-estrogen. It targets the estrogen receptor. So it works in a fundamentally different way than than other drugs that currently work. It is intended for patients who are estrogen receptor positive. So currently we already have drugs that work some for patients who are estrogen receptor positive. However, those drugs initially work, and then afterwards those drugs stop working.
So, what's different about this one, about the mechanism then?
So the mechanism, it's a fundamentally different mechanism from the other drugs that are commercially available, that FDA approved. This targets the way the estrogen receptor functions. So in order for estrogen receptor to function, it functions by binding to certain coregulator proteins. And what this does is it blocks the interaction between the estrogen receptor and critical coregulators. So it's a fundamentally different mechanism than other drugs that are commercially available.
We've been hearing here in San Antonio about the way anti-estrogen therapies can modulate the environment and perhaps influence the development of the disease. How would yours differ? Is it just another agent?
Well, it's not, but one of the key things is, in order for resistance to happen, resistance happens by mutations, alterations in the estrogen receptor, but at the same time, one of the key ways all those mutant receptors work is that they interact with coregulators. This cuts out the legs out from underneath the estrogen receptor. It prevents the estrogen receptor from interacting with a mutant or wild type, from interacting with a coregulator, so it's fundamentally different.
Sounds a great idea. What did you do in practice in your preclinical work?
So we first developed an agent and we've tested it in a variety of breast cancer cell lines first. Then we tested it in animal models. And interesting, we also tested it in primary breast cancer tissues, so tumor tissues from patients with breast cancer. We tested whether those are exactly the patients who would benefit from the therapy, and we showed that in the tumor tissues that this drug has activity.
Okay, activity. What about potential toxicities too?
So that's a great question. Anytime you develop these drugs, is a question of toxicity. So we tried to establish what's called a toxicity to therapeutic ratio. So you go 100 times higher dose than what you would give the animal for therapeutic efficacy and try to see this toxicity. And even at that higher dose, we did not see toxicity in the animal.
So what do you think is the big advantage of going for the co-regulators?
I think when you target co-regulators, resistance is a little harder to come by. So you're targeting a broad panel of interactions. In order for resistance to develop, the receptor must change in a fundamentally different way than what we've seen it develop so far in order to avoid the activity of this drug.
Right, so what's come out of the study so far that is pointing in a clinical direction?
So we have a lead candidate that we have funded to take it forward to IND, or investigational new drug status. So we're currently performing the IND enabling studies and bringing it forward to clinical trials. What
do you think doctors should take home from this sort of promising but as yet unproven research?
Well, this is a novel way to target the estrogen receptor. And I think this highlights additional ways to target the estrogen receptor. We're still targeting this estrogen receptor, but in a more effective manner.
So the status of ECBI at the moment is what?
It's preclinical, rapidly heading towards final testing for IND status, and then trying to move it forward to the clinic.